Today we are going to learn UV visible and fluorescence spectroscopy. These two techniques are very important techniques in nanomaterial characterization. In today's lecture, we will be learning about what is spectroscopy, and we will also learn what is the working principle of UV visible and fluorescence spectroscopy, both theoretically as well as practically through demonstration. Let us see what is spectroscopy. Spectroscopy is the study of the interaction between electromagnetic radiation and matter. It involves analyzing how a substance absorbs, emits, or scatters light to provide information about the composition, structure, and physical properties of substances. There are various types of spectroscopic techniques like absorption spectroscopy, emission spectroscopy, and scattering spectroscopy. In today's lecture, we are going to learn about UV visible spectroscopy and fluorescence spectroscopy in detail. Let us briefly learn the history of spectroscopy. In 19th century, Dr. William and Dr. Joseph made a groundbreaking discovery of absorption spectra. In 1941, the first commercial UV visible spectrophotometer was introduced. Currently, the spectroscopic techniques are a standard analytical tools for real-time and high precision measurements. Let us see what is UV visible spectroscopy. UV visible spectroscopy is a versatile analytical technique which is used to measure the absorption of light by a sample in the ultraviolet and visible range of the electromagnetic spectrum. So, that is why it is called as UV visible spectroscopy. It can measure the absorption of light by a sample in both ultraviolet as well as visible range of the electromagnetic spectrum. And some spectrophotometers have an extended wavelength range. It can measure even in the near infrared range. So, that is from 800 to 3200 nanometer. And the measurement is based on the absorption of light due to excitation of electrons in molecules from lower to higher energy states. Let us see the principle of this technique. The principle is when light passes through a sample, some of the energy is absorbed by the molecules in the sample. The electrons in the molecule get excited and move from lower energy level, that is from ground state, to higher energy level, that is the excited state, by observing energy of a particular wavelength. Let us learn about transmittance and absorbance. Transmittant is defined as the ratio of the transmitted intensity and the incident intensity. It has a value between 0 and 1. And absorbance is the measure of amount of light absorbed by the sample at a specific wavelength. The transmittance and absorbance are inversely related and have a logarithmic relationship. For example, if I have an absorbance value of 0, that means 100% transmittance, and if the absorbance value is 1, that is 10% transmittance. Let us learn about Beer-Lambert law. The Beer-Lambert law is a linear relationship between the absorbance, concentration of the analyte, molar extinction coefficient, and optical path length of a solution. If you are using a sample with higher concentration, that leads to greater absorption, which reduces the transmittance. And if you are using the longer path length, that increases the interaction with the light, leading to more absorption. Longer path length refers to the distance that light travels through the sample in the cuvette or cell. This path length is typically measured in centimeters, and the molar absorptivity is the intrinsic property of a particular chemical species. And molar absorptivity is a measure of how strongly a substance absorbs light at a particular wavelength. Let us learn about the parts of spectrophotometer. The first one is light source, which provides the radiation, and we have two light source. One is a deuterium lamp, which is for UV range, and we have tungsten lamp. This is for the visible range. I will explain about this light source in my next slide. And we have monochromator, which separates the light into its component wavelength and selects the desired wavelength. And here we have a sample holder which holds the sample. We can use the quartz cuvette if you want to measure your sample in UV range, and you can use the glass cuvette for visible range. And in some cases, we can also use the microplates for high throughput analysis. For example, if you want to have a multiple samples like a 96 well plate, you want to analyze all the samples simultaneously. For that, we have to go for microplate holder. Then we have the detector, which measures the intensity of the transmitted light after passing through the sample. Then we have the processor unit, which converts the signal from the detector into absorbance or transmitted values. Then we have the display unit which displays the data as spectra, tables or graphs. And in this picture what we are seeing is the double beam UV visible spectrophotometer. So, the advantage of using this double beam UV visible spectrophotometer is we can measure the observance of sample as well as reference simultaneously, and most of the lab they use this double beam UV visible spectrophotometer. Let us see the details of light source. The first one is deuterium lamp. So, the deuterium lamp is mainly useful to measure the observance of sample in UV range, that is 200 to 400 nanometer. The deuterium lamp uses arc discharge from deuterium gas. So, the one drawback is it has a short half life. So, we have to frequently replace the deuterium lamp. The next one is tungsten halogen lamp. It is mainly useful to measure the observance of a sample in visible range, that is from 400 to 800 nanometer, and the filament emits lights when heated. So, that is why before we perform the experiments we have to switch on the equipment and allow some time so that the lamp get warm up, and it has a very low noise and low drift, and it has a longer life. And third one is mainly in the near IR region, that is the xenon lamp. So, it can measure your sample from UV to near IR region, that is from 185 to 2500 nanometer. It emits short flashes of high energy light, and it is mainly useful for photosensitive materials, and here there is no need for warm up time of the lamp. Let us learn about the cuvette. The cuvette for UV visible spectroscopy is done based on the factors like material transparency, path length, volume and chemical inertness. The quartz cuvette is mainly used for measuring the observance of sample in the UV range. The reason is the quartz cuvette is transparent to UV radiations, and the glass cuvette is mainly used to measure the samples observance in the visible range, because the glass absorbs the UV radiation. So, that is why we have to use the glass cuvette for measuring the observance in the visible range, and we have to use the quartz cuvette for measuring the observance in the UV range. And these cuvettes are usually frosted on two opposite sides to increase the accuracy of observation measurements. You can see here. So, two sides are opaque and two sides are transparent. The cuvettes are also supplied as matched pairs which have identical optical properties. So, this minimizes the variation between the sample and reference in a double beam spectrophotometer. So, the cuvettes are always available in pairs to avoid the batch to batch variation as well as to minimize the variation between the sample and reference in a double beam spectrophotometer. Let us see the difference between single beam and double beam spectrophotometer. In single beam spectrophotometer, it uses one light path, and reference and sample are measured separately. Whereas, in case of double beam spectrophotometer, it splits the light into two paths for simultaneous measurement of sample and reference. And the measurement process require manual switching between the sample and reference in single beam spectrophotometer, whereas in case of double beam spectrophotometer, it measures the sample and reference simultaneously. So, that is the advantage of this double beam spectrophotometer, and speed wise it is slower due to separate measurements, and it is faster as both paths are measured in real time, and accuracy wise it is more prone to fluctuations in light source intensity, whereas in case of double beam spectrophotometer it compensates for light source fluctuation and provides a better accuracy. So, that is why most of the lab they use these double beam spectrophotometer. Let us learn about the importance of blank and baseline calibration. The blank calibration eliminates the instrumental noise, and when we measure the absorption of the sample at a particular wavelength, the blank calibration ensures that the observance of the solvent is subtracted from the final reading, and this way it improves the measurement accuracy. The next one is baseline calibration. So, when we do the spectral scan of the sample, the baseline calibration basically subtracts the absorption spectrum of the solvent used for making this sample solution to improve the quality of the obtained data. So, it is similar to how we zero the weight of the container when weighing something. For example, whenever we go to the fruit markets, so the vendor remove the weight of the basket before he put the fruits. So, that means we are getting the exact weight of the fruits, not with the basket. Similarly, when you are using this baseline calibration or the blank calibration, it will remove the absorbance of solvent and it will provide only the absorbance of your particular sample. Let us see some examples for understanding how we can use this UV visible spectrophotometer for characterizing the metal nanoparticles. The first example is gold nanoparticles. When you use the gold nanoparticle, you get a single peak, and when you are using these gold nanorods, you get two peaks. One is for transverse plasmon band and the other one is for longitudinal plasmon band. Let us see another example for understanding how we can use this U-visible spectrophotometer for measuring the absorbance of silver nanoparticle. And in this figure you can see that, so this is a colloidal silver in various stages of aggregation. Usually the silver nanoparticle will be in yellow color, and to get the proper silver nanoparticle we have to use the metal precursor and the reducing agent, that is sodium borohydride, in the right ratio. If you are using the ratio of 2, you will get a highly stable silver nanoparticle. If the ratio is 2.1, the nanoparticle is stable for 30 minutes. If the ratio is 1.9, it is stable for 20 minutes. If the ratio is 1.8, the silver nanoparticle is stable only for 5 minutes. You can see here with respect to the aggregation, the color is also varies, because as the particle size is bigger with respect to the aggregation. And when we measure the observance of silver nanoparticle using the U-visible spectrophotometer, so you get the peak around 400 nanometer for silver nanoparticles. And based on the previous literature, we can get an approximate idea about the size. For example, if you are getting a peak around 420 nanometer, the size is between the 35 to 50 nanometer. And the size of the silver nanoparticle can be confirmed by using the transmission electron microscope. Let us see the applications of U-visible spectroscopy. We can use it for characterization of nanoparticles, as we learnt in the previous slides. Like we can use it for studying the metal nanoparticles, so as it exhibit SBR peaks in the U-visible spectrum, and we can use this spectroscopy for understanding whether the nanoparticle is in the dispersed state or in the aggregation state. And we can also use it for measuring the concentration. Nanoparticle concentration in a colloidal solution can be measured using the spectrophotometer. For example, we can calculate the silver nanoparticle concentration using its extinction coefficient at 400 nanometer, and we can also use it for analyzing the size and shape. So, this U-visible spectra can provide insights into the nanoparticle size and shape. For example, if you are having a rod shaped nanoparticles, as I told you, you get two peaks, one for the transverse, one for the longitudinal, and we can also use it for understanding the optical properties. So, the optical band gap of semiconductors and quantum dots can be also determined. So, we can measure the band gap between the conduction band and valency band. We can also use this in aggregation studies to monitor the changes in U-visible absorption peak to evaluate the nanoparticle stability. For example, if the nanoparticle is aggregated, there is a redshift or broadening of peaks. So, that indicates the aggregation of nanoparticle. Usually for gold nanoparticle you get a sharp peak. If the nanoparticle is aggregated, you get the peak shift as well as the broadening of peaks. And we can also use it for drug release studies. It measures the drug release kinetics from nanocarriers like liposomes or polymeric nanoparticles. For example, if you are loading your nanoparticle with the anti-cancer drug, or if you are loading with drug like curcumin, so we can measure the absorbance of the particular drug. In case of curcumin, we can measure the absorbance at 425 nanometer. So, when you are using this curcumin encapsulated nanocarrier, with respect to time, we can understand the release of drug by using this U-visible spectrophotometer. And we can also use it for understanding the functionalization, that means the addition of functional group to the nanoparticle. So, it confirms the binding of biomolecules like proteins or DNA to the nanoparticle surface. For example, you are having a positive charge nanoparticle and your DNA is negative charge. So, once the nanoparticle attached to the DNA, then we can see the changes in the SPR peak, and we can confirm the binding of biomolecules to the nanoparticle surfaces. We can also use the U-visible spectrophotometer for studying the photocatalytic activity of nanomaterials. For example, here we can use these TaO2 nanoparticles for the photocatalytic degradation of methylene blue dye. So, that can be monitored by measuring the absorbance decrease at 665 nanometer. And when we are using this kind of dyes, methylene blue, we can use the disposable cuvette, or if you are using the glass cuvettes or plastic cuvettes, make sure that the cuvettes are cleaned properly. Otherwise, that will interfere with the next sample. Let us see some of the common issues which we face when we use the UV visible spectrophotometer for measuring the absorbance of sample, and also how to overcome those challenges. The first problem is sample preparation issues. So, the possible cause may be the contamination or improper mixing. We can use the uniform volumes, or we can filter the samples to remove the impurities or contamination and make sure that there is a proper mixing. And if you are facing a problem like noise, so that may be due to the instrument instability, or it may be due to the cuvette which is not clean, or it may be due to the environmental interference. So, calibrate the instrument, clean the cuvettes, and make sure that there is a vibrationless environment. And if you are facing a problem in the blank or zero calibration, so the possible cause may be due to the contaminated blank, or it may be the cuvette is not clean, or there is no zeroing. So, always use the fresh blank, clean the cuvettes as I told earlier in the previous slides also. So, always zero the instrument. And if you are facing the problem like a too high concentration, so when the sample concentration is too high, it exceeds linear range and deviates from Beer-Lambert law. So, in this case we have to dilute the sample or use shorter path length cuvettes. I hope you got the overall idea about the UV visible spectrophotometer. The next technique what we are going to learn in today's lecture is fluorescence spectroscopy. The fluorescence spectroscopy is an analytical technique used to measure the fluorescence emitted by a sample when it absorbs light. The fluorescence is a type of luminescence which occurs when a molecule, that is a fluorophore, absorbs light at one wavelength, that is the excitation wavelength, and then re-emits light at a longer wavelength, that is the emission. And this fluorescence spectroscopy is widely used in chemical, biological and environmental analysis. Let us see the principle of fluorescence. So, the fluorophore absorbs the light and causing an electron to jump to a higher energy level, that is the excited state. The molecule loses energy through internal conversion or vibration and returns to a lower energy excited state, and when returning to the ground state, the molecule emits photon of lower energy. So, that is the fluorescence. Let us learn about the parts of fluorometer. In the fluorescence spectrophotometer, we have this light source, that is a xenon flash lamp. So, it provides energy needed to excite the sample. Followed by that, we have this excitation monochromator. So, it selects the exact wavelength of light which is used to excite the sample. Then we have the sample holder. Usually we use the quartz cuvette, transparent on all four sides. And we have the emission monochromator that selects the emission wavelength similar to the excitation monochromator, but for the emitted light. Then we have the PMT detector, that is a photo multiplier tube, and which can amplify the light and helps to detect even the faint signals. And this detector is kept at 90° to avoid the interference of the transmitted light. So, when you have this light source and your cuvette is here and the detector is placed 90° to your light source. So, in this way it can avoid the interference of the transmitted light. And we have the computer and software which can collect and analyze the data to produce the fluorescence spectrum. Let us see the working of the instrument in detail. The light source emits light which is then passed through an excitation monochromator. Then the excitation monochromator selects the desired wavelength. The excitation light then illuminates the sample, exciting the electrons in the fluorophores to a higher energy state, as I explained in the principle of the fluorescence. So, the fluorescence light is emitted when the high energy electrons return to their ground state. The emitted light is of higher wavelength because of the vibrational energy loss suffered by the electrons. The emission passes through a monochromator which filters out the emission wavelength. The emitted light is detected by the detector and the fluorescence spectrum is recorded, which shows the intensity of the emitted light at various wavelength. Whenever we synthesize fluorescent nanomaterials, we have to calculate the quantum yield. The quantum yield is one of the important parameter in deciding the fluorescent nanomaterials for various applications. So, let us learn how we can calculate the quantum yield. So, the fluorescent quantum yield is the ratio of the number of photons emitted by a fluorophore to the number of photons observed. It measures the efficiency of fluorescent emission, and it is a key factor in determining the brightness and sensitivity of fluorophores in applications like fluorescent microscopy, diagnostic and sensor. So, that is why whenever we synthesize the carbon dots or quantum dots, we have to check whether it has a high fluorescent quantum yield or not. So, if it has a high quantum yield, so that makes the carbon dots bright and sensitive. So, this is one of the essential parameter for applications in biomedical field, especially in case of bioimaging and biosensing, where strong fluorescent signals are required to detect low concentration of analytes. The quantum yield can be calculated by comparing the fluorescent intensity of a sample to a reference material. So, usually most of the lab they use this quinine sulfate, or we can also use the fluorescein with the known quantum yield. So, the quantum yield of these two chemicals are already well known. So, we can use this as a kind of standard reference material, then we can calculate the quantum yield for our sample. The first step is the solutions are prepared for both sample and reference in the same solvent, such that the observance values at excitation wavelength for both the solutions are less than 0.1. So, this is done to eliminate the inner filter effect, where the sample observes the emitted light due to high concentration. And the next step is the fluorescence spectrum is recorded at same excitation wavelength for both, and integrated area under the fluorescence spectrum is calculated to calculate the quantum yield of the particular nanomaterials. And we can use this equation to calculate the quantum yield. Let us see some of the nanotechnological applications of fluorescence spectrophotometer. The first one is we can use this fluorescence spectrophotometer for characterization of fluorescent nanomaterials, for assessing the optical properties and quantum yield of nanomaterials such as carbon dots or quantum dots. And we can also use it for biosensing and diagnostics. If you are having a fluorescent nanomaterials, so it can be act as a kind of fluorescent nano sensor. So, once it bind to the particular cancer cell, and it can glow. So, based on that we can use it as a kind of specific fluorescent nano sensor for detecting the specific diseases, and we can use it for study of energy transfer. So, it can be useful for application in detecting the molecular interaction and conformational changes. If there is any conformational change, so the fluorescent intensity will go down or it can go high. So, based on the intensity we can understand the conformational changes. We can also use it for nanostructure surface studies, for studying the surface interaction between nanoparticles and biological molecules using fluorescence quenching or enhancement. For example, if you are having a fluorescent nanoparticle and once it binds to the particular analyte, these nanoparticles can give fluorescence or the fluorescence can be quenched. So, that means the fluorescence can be on or the fluorescence can be off. So, by this way we can understand the surface interaction between nanoparticles and biological molecules. We can also use it for studying the photocatalysis. For example, we can use it for evaluating the efficiency of photocatalysis like doped nanoparticles under light exposure. When you are using the light, and we can see that whether the photocatalytic efficiency is improved or not. And we can also use it for nanotoxicology, for detecting the release of ROS, that is the reactive oxygen species, by using the fluorescent based assays such as DCFH DA assay. If ROS is more, then fluorescent intensity will be high. No ROS, then no fluorescence. With respect to the fluorescence, we can quantify the ROS. Let us see some of the common issues which we face when we use the fluorescence spectrophotometer and how to overcome those challenges. The first problem is weak or no fluorescence. So, it may be due to the low fluorophore concentration or it may be due to the incorrect excitation wavelength. So, we can overcome by increase the concentration, or we can check the absorption spectrum and use the correct wavelength. And next one is high background signal. It may be due to the solvent fluorescent or impurities, or it may be due to the improper blank, or it may be due to the contaminated cuvettes. So, we can use the high purity solvents, or you can subtract the blank spectrum from the sample spectrum, or you can use the clean cuvettes. And third one is the fluorescence quenching. So, the fluorescence quenching may be due to the presence of quenches like oxygen or heavy metals, we can remove the oxygen by purging with nitrogen or using deoxygenated solvents. And it may be due to energy transfer to nearby molecules. So, we have to identify and remove the potential quenches from the solution. And the fourth one is inner filter effects. It may be due to the high sample absorbance. So, we always have to dilute the sample to maintain the absorbance, or it may be due to the pore calibration. Calibrate the instrument regularly using standard fluoropores. And next one is photo bleaching. So, the photo bleaching may be due to the prolonged excitation light exposure. So, you can use reduced light intensity, or you can use a pulsed light source or switch to more stable fluorophores. So, the last one is the temperature effects. So, this may be due to the temperature fluctuation. So, it can be overcome by maintaining the constant temperature condition throughout the analysis. Hope you got the overall idea about the fluorescence spectrophotometer. Let us go to the lab and learn this technique more in detail. Now, we are going to learn UV visible spectroscopy. For this, we have already synthesized two materials. One is gold nanoparticles synthesized using sodium citrate, and another one is tannic acid capped gold nanoparticles. We will need a set of quartz cuvette, ultra pure water for preparing dilution, tips and pipette, tissue paper, and beaker for discarding the waste. First, turn on the machine and wait for the lamp to be warm up. Then set the machine on a spectral scan and keep the wavelength from 400 to 800 nanometer. Select a step size of 5 nanometer. First, we have to calibrate the baseline. For that, we have to use ultra pure water. While adding any sample, hold the cuvette at an angle to avoid any bubble formation in the cuvette. Now place the cuvette in the machine. Ensure that the clear sides of the cuvette in line with the light source and detector. Now we have to select the blank calibration option. You can see that the machine will take a spectral scan from 800 to 400 nanometer, which will be the baseline for our measurement. After the calibration has been done, we will take a blank reading of the solvent that we have used for the preparation of gold nanoparticles. In this case, it is ultra pure water. So, we will place the cuvette in the machine. And press the 0 option on the software. After blank reading, discard the blank solvent and add 2 ml of gold nanoparticle into the cuvette. The cuvette holds approximately 3 ml of the sample. Here, first we are going to take UV reading for gold nanoparticles synthesized through Turkish method. You can see the ruby red color of the gold nanoparticles. Add the sample to the cuvette and place it carefully, ensuring proper orientation. Now press start to initiate the measurement. You can see that the peak is going above 1 absorbance. So we will dilute the sample to get a reading in the proper range. So we will take out the cuvette and remove 1 ml from it and store the extracted sample back in the vial. Now add 1 ml of ultra pure water to the cuvette to achieve a half dilution of the gold nanoparticle sample. Mix it properly and ensure there are no bubbles in the cuvette, and place it in the cuvette holder facing the light source and detector. Once again press on the start and take the reading. Now you can see that after dilution the peak intensity has decreased and the gold nanoparticle synthesized through Turkvich method is showing a peak around 520 nanometer. Now we will take the observance for tannic acid capped gold nanoparticles. First we will discard the previous sample and clean the cuvette using ultra pure water. Gently tap the cuvette on the tissue paper to dry it. And then add 2 ml of the gold nanoparticle sample to the cuvette. Since the solvent is same for this sample also, we are not repeating the blank calibration. Place the cuvette in the machine and press start. You can see that it is taking a reading, and you can observe a broad peak around 530 to 550 nanometer. This shift in the peak towards right side is due to the capping of tannic acid on gold nanoparticles. For saving the file, you can click on the Save As option and save the file with proper sample name and date. In this demonstration, we are going to perform fluorescence spectroscopy to analyze the photoluminescence spectrum for the 2 carbon dot samples that we have synthesized in one of the previous lectures. The first carbon dot sample was synthesized using the hydrothermal method and the other one using a microwave assisted method. The materials required are a beaker to discard waste, a 4-sided quartz cuvette, a wash bottle, tissue paper, microtips and micropipettes. First, switch on the machine and wait for 15 to 20 minutes for the light source to warm up. Once the warm up is done, open the software and select the spectrum option, and within that select the emission option. For the first carbon dot sample synthesized using the hydrothermal method, the excitation wavelength is around 270 nanometers. So we will set the emission wavelength at 290 nanometers and the end wavelength for the spectrum at 700 nanometers. Add 2 ml of the carbon dot sample into the 4-sided quartz cuvette. Ensure that the cuvette is clean and there are no bubbles in the sample. Now place the cuvette inside the sample holder of the machine. After closing the chamber, press run to start the PL spectrum measurement. You can see that it has started collecting the emission data from the sample. To save the file, we will enter the sample name and click OK to save it at our desired location. You can see the PL spectra on the screen for the carbon dots synthesized using the hydrothermal method. Now for the next carbon dot sample synthesized using the microwave assisted method, the excitation wavelength is around 280 nanometers. So we will set the emission wavelength at 300 nanometers and the end wavelength of the spectrum at 700 nanometers. Discard the previous sample from the cuvette and wash it 2 to 3 times using distilled water. ensure that the cuvette is completely clean. Dry the cuvette using tissue paper. Fill 2 ml of the carbon dioxide sample in the cuvette. Ensure that there are no bubbles in the sample and the cuvette is clean. Place the cuvette in the sample holder. Press run to measure the PL spectrum. You can see the PL spectrum for the carbon dots synthesized using the microwave assisted method on the screen. There is an extra peak in the spectrum due to the lower purity of the synthesis in the microwave method, but it has more fluorescent intensity than the hydrothermal method. After taking the measurement, save the file, exit the software and turn off the machine. As a summary, in today's lecture, we learned the principle of UV visible and fluorescence spectroscopy, and we also learned how to use the UV visible and fluorescence spectroscopy for characterizing the nanomaterials both theoretically as well as through practical demonstration. Thank you for your kind attention. I will see you in another interesting lecture.